polymerase that attaches to the promoter there, if it could do so, would not be able to run along and transcribe that gene. The methyl groups get in the way of RNA polymerase as it tries to go along the gene. So the entire length of the gene is going to be messed up if the methyl groups are attached to it. And that will permanently shut those off, whether it's in the heterochromatin form or not. Does that make sense to everyone? By an enzyme. And the enzyme has to recognize specific sequences within the stretch to methylate. So again, it all comes down to something interacting with a sequence of nucleotides. You don't just randomly attach methyl groups. They're attached very specifically at strategic points.
So I presume the only way to remove them would be through another enzyme then?
Yes.
Okay.
Typically when we methylate, we don't ever demethylate them. Once they're methylated, that's forever. Which is why things like nerve cells can't reproduce, because we've methylated the genes that allows for the reproductive mitosis process in nerve cells. But if you could actually re-engineer or reintroduce an enzyme that can recognize this location, and you can demethylate that gene, then you can allow that gene's function to carry out in that tissue. And that's one thing that's happening in biotech right now, is try to reactivate genes of use in tissues that are damaged. And one way to do that is to demethylate. But it's not easy to do. We need to find the right enzymes. And you can't demethylate the wrong regions. It must be a targeted area to make sure that's the only gene you're activating. So when